Pancreatic cancer has an extremely poor prognosis, with only 12% of patients surviving 5 years post-diagnosis. This makes pancreatic cancer one of the deadliest cancers, with over 500,000 new cases diagnosed worldwide each year.
Pancreatic cancer is often diagnosed at an advanced stage, typically only after metastases have developed, making it particularly challenging to treat. Consequently, the only chance for a cure is through surgery combined with chemotherapy.
Alexander Kleger, a physician-scientist and stem cell researcher from Ulm, is a leading expert in pancreatic cancer research. He aims to investigate these aggressive tumors using entirely new approaches and biological models, with the goal of advancing scientific strategies to combat them.
We have concepts, but they remain just ideas for now. It is important to stress that. A new approach is the development of organoid systems derived from pancreatic carcinomas. An organoid system is a type of cell structure that replicates the function and morphology of an organ or tumor.
Alexander Kleger's team was among the first to successfully grow organoids from both healthy pancreatic tissue and tumors using human pluripotent stem cells. Using these three-dimensional cell structures, scientists can observe their development, communication, growth, as well as diseases, almost like watching the progression of a pancreas and its cancer develop. This approach allows researchers to study the early stages of cancer across various forms.
We believe that the key advantages of each organoid system are twofold. First, they more accurately recapitulate the heterogeneity of the original tumor as it developed in the patient. Second, we can adapt the organoids to our needs, which is the primary focus of our work. This adaptability allows us to artificially modify the organoids, enabling us to recreate the early steps of tumor development.
Genetic tools such as CRISPR-Cas, which allow for precise modification of the cells within organoids, play a crucial role in this process. Alexander Kleger is optimistic that these molecular biology methods will significantly improve patients' chances of recovery.
The aim is to utilize these organoid systems to identify biomarkers that can detect cancer at much earlier stages, allowing for more effective and efficient treatment with existing methods such as surgery and chemotherapy.
Kleger and his team have developed an innovative method to culture human organoids outside of traditional petri dishes, while also circumventing the need for animal experiments. They grow the organoids on pig bladders. These complex structures are easily observable under a microscope. But why pig bladders?
The advantage of using pig bladders is that studies have shown their matrix partially resembles that of normal human pancreatic tissue. We conceived the idea of removing the cellular components, specifically the epithelium, and then transplanting the organoids onto this matrix for further cultivation. This method has also been used for substrate testing, revealing benefits at multiple levels. Furthermore, we demonstrated in direct comparative studies that this approach allows us to reduce the use of mice in animal experiments.
Using organoids derived from a particularly aggressive type of pancreatic cancer, Kleger's team employs a pipetting robot to test substances that specifically target and kill tumor cells. This approach allows doctors and researchers to explore effective strategies for combating cancer in the future using targeted therapies. But there is still a long road ahead. The development of pancreatic organoids from stem cells alone took almost 10 years. Nevertheless, Kleger remains optimistic about future progress. Using a range of modern cell and molecular biology techniques, he aims to achieve his goal of providing individualized diagnosis and treatment for every patient affected by pancreatic cancer.
I am convinced that personalized medicine will be essential for successfully treating pancreatic cancer. The heterogeneity of this tumor disease is so significant that each patient's tumor must be regarded as a distinct condition requiring tailored treatment.